placer contar con su presencia. Y sin más preámbulo, bueno, les voy a dejar con la doctora Connie, quien quiere hacer algunos comentarios más sobre la doctora Yana, para introducirnos con nuestra ponente del día de hoy. Gracias.
Thank you so much for having me here. You guys have a really beautiful university, and I'm honored to be invited to come down and talk with you today. Is it possible to turn the lights in the front down? The slides will look better. Or possibly, yes. Sorry. So, I'm Jessie, and I'm a postdoc at UC Berkeley. You see there's a UCSF here too, so I spend my time across the Bay Area at UC San Francisco and UC Berkeley. But that's correct that I'm a postdoc. And I wanted to add that I'm starting a laboratory at Oregon State University. So, we had a great conversation just before this about Mycoavidus systematics. And if that or anything else resonates with you and you'd like to collaborate, please contact me. Okay, so today we're going to talk about fungal symbiosis genomics. And fungi actually form a huge diversity of symbioses. So I'm going to talk in particular about fungal bacterial interactions and fungal plant interactions. But these symbioses impact us every day. So examples of fungal bacterial symbioses include producing beautiful food products like breads, sourdoughs, cheeses. The blue cheeses in particular are fungal bacterial products. Fermentation like beers, especially if you like sours. And of course antibiotics. So I think everybody in the room has probably benefited from the results of fungal bacterial symbioses. But sometimes it's uglier than that. So sometimes fungi are very clever and they've figured out how to infect humans. So they can degrade skin and get into hair. They can infect the lungs and often kill humans. They also form symbioses with plants. So bacterial, fungal, and plant fungal symbioses are what we'll talk about today, but almost all of the land plants and many of the cyanobacteria are in symbioses with fungi that live in their tissues, in leaf tissues, and also on their roots. So this broad diversity of fungal symbioses really gives us a chance to ask in this genomic area, what are the molecular impacts of these fungal symbioses, and how can we use fungal genomes to figure out what the basis of these interactions are and how this has shaped evolution of these symbioses. So to ask this question, we're going to use a systems biology approach, where we use a series of genomic approaches, and I wanted to just walk through these. So some of these will be obvious, like microscopy is microscope, but what is genomics? Of course, in the cell we start with DNA, and that is transcribed RNA or messenger RNA and proteins. And we can quantify the level of each of these products in different settings and then infer from their differential abundance which genes or proteins or metabolites are important. And we can take it to another level and say, okay, here's the cell. We have DNA, RNA, and protein, but we also have metabolites and volatiles. So we can quantify each of these using different techniques and then say one is more abundant here and not here, and therefore it's implicated in this process. So I'm going to use these color codes as a little key. They'll go down to the corner, and the kind of data that I'm talking about will be in a larger hexagon, just to keep us all on track. Okay, so I have two stories for you today. The first one's about tropical rainforest mushrooms, and the second one is about fungal endosymbionts, or bacteria that live inside of fungal cells. And we'll start in the rainforest. So these are the rainforests that are distributed around our world. You might notice that some, including the area where I live, is actually temperate rainforest, but we're here to talk about the tropical rainforest. And I've worked the most in northeastern South America and Guyana, and also central West Africa and Cameroon. So I'm going to focus on those. And you may notice, and very well know, that these two land masses used to be together. So they fit together like a jigsaw because they used to be together. And we know this from several lines of evidence. So one is the shapes. They perfectly fit together. But the other is that the biodiversity that's present on both continents, especially in northeastern South America and central West Africa, is really similar. So in other words, you have similar plants that host similar fungi in both locations. Okay, so just a brief refresher for the folks who were in the last seminar. This will be a review for you. This is a fungal life cycle. So when we think about fungi, oftentimes we think about mushrooms, and that's correct, but this is actually a reproductive structure. So this organism spends the majority of its time in this asexual phase we call mycelia. These are long, you could think of like a hair, a long cell, and they live in the soil. In this case, they're associated with plant roots, so wrapped around plant roots. And when they fuse, they form this reproductive structure, and then on the underside of these gills of the mushroom are these cells called basidia. And the basidia are where the spores are produced. So there's meiosis and mitosis happening, and then these spores are reproductive propagules that go on to germinate into hyphae and then form a mushroom. So hold on to those two terms, the mycelia and the basidia, because they'll come back. Now the reproductive tissue, or the hymenium, where the basidia are born, covers the underside of gills like this, but there are many morphologies of fungi, especially in the tropics, and that tissue could cover pores, like the bolete-related fungi. It could be smooth, it could be wrinkles. Here's a gastroid fungus, like a truffle. And the one that I know the most about and that I'll talk about today are the coral fungi in the genus Clavulina. So in this case, you have a tree-like structure with the fat end sticking into the soil, and then the branches come up, and they're surrounded with the fertile tissue or hymenium where the spores are being produced. So these are symbiotic fungi. They're in symbioses with plants. This is an equal trade whereby both partners benefit. The tree is doing photosynthesis, of course, and making sugars or carbohydrates from carbon dioxide. And it's passing those sugars down through the trunk, through the tree roots to the fungi. The fungus, meanwhile, is out in the soil with its filamentous hyphae, sucking up phosphorus and nitrogen and other nutrients and passing them back to its plant hosts. And they both benefit from being together. In fact, so much so that they've co-evolved, and we can see signatures of their co-evolution in their genomes. So here in Guyana, right between Venezuela, Brazil, and Suriname, we have many study sites, and we spend the most time in the Pataro study site. It's just there. And the host that we think the most about is the Simbi corymbosa. So these are beautiful trees. They make a huge biomass. There's a human there for scale, and they have this, And they have this reiterative growth where they have multiple fused stems, and it's a beautiful tree. But it's also a huge habitat for these ectomycorrhizal fungi that are associated with the root tips. So you have to remember when you see a tree like this that's just massive, that there's just as much tissue and space for microbes to live below ground. And if we think about an individual root tip, it looks something like this. So this is a scanning electron micrograph of a root tip that's been colonized by an ectomycorrhizal fungus. And I false colored it. It doesn't look like this in real life, right? With the plant tissues, they're in purple inwards, and the fungal hyphae are red. So you can see the filaments or hairs are wrapped really tightly around these roots and in fact between the cortical cells, and their contact point is where they're sharing these sugars and nutrients back and forth. Okay, so here's some of the diversity in the genus Clavulina. These are mostly from South America. There are a few from Africa, including this one and this guy. But the takeaways are that you have this coral shape mostly, right? And you have a demarcation where sporulation is happening up here. This end is the part that's in the soil. And then we have some interesting morphologies, like some of them are funnel shaped, others are reduced or restupinate, and we're interested in evolutionary patterns between them and relationships between them. But I just wanted to present this question. Let's say that you're walking through the forest and you see this beautiful fungus. How can we collect this and identify it and begin to study it? And in the field, this starts with really a color match. So we bring these color guides. This is something that an artist might use. And you have a high resolution color swatch and you say, okay, I think it's about this color on the base and this color on the tip. And basically use your eyes to note everything you can. How big is this mushroom? How wide is it? How frequently does it branch? How does it smell? How does it taste? And we're also interested in the color of the spores that are produced. So for Clavulina, the spores are often white in deposit. These have been created from fungi with gills, but the concept is similar. Spores come in all sorts of colors with different morphologies that you can see with the microscope. So that's part of the identification process. Then we dry these fungi. Remember, we're in the tropics in the rainforest, so we use silica gel beads to dry the fungi. This is something you might find if you buy like new pants or new shoes. Same thing. So you take the mushroom and put it in a Tupperware with the silica gel beads, and then you have a dried specimen that's ready to transport back to lab. It's not going to rot and sort of stop the decay process. once we get back, then we're interested in using DNA for molecular studies. We've been sequencing genes, and we're hoping soon to move to genomes. And of course, microscopy. So using a microscope to look at fungal cells, including basidia, for dimensions and special features that we can use to identify species. So these basidia, I'll just remind you, come from the fertile tissue, so the underside of the gills of these mushrooms. And this is, again, just reproductive, asexual. So these basidia, especially for Clavulina, have a few characters that I'll just point out. So these are more scanning electron micrographs of Clavulina species from Guyana. And you can see that mostly they have 2, sometimes 3, but they're, these horn-like structures are called sterigmata. This is the spore on top of the sterigmata. So we have 2 curved or cornuate sterigmata and these big globose, smooth spores. They don't have any sort of warts or ridges. These ones are dried, so they're a little wrinkled, but that's what they look like under the scope, and we use these to say, yes, for sure, Clavulina. So again, the features are that the spores are nice and round and smooth. You can see a big oil droplet in the middle. It looks like a big green blob. And then the basidia themselves have these 2 incurved or cornuate sterigmata. Okay. So here's some of the species that I've described in my career. I have many others that are undescribed too, so again, if there's any interesting collaboration, and it sounds like there is, we should definitely hatch a plan to work together on this. But I just wanted to point out some of the morphological diversity in the group. We have different colors, different sizes, different branching patterns, and oftentimes the name will reflect the way these fungi look. So look, here's a Latin for pink. I think this one's probably my favorite. Sometimes the name will be about where these fungi are from. So this is Guyanensis, the lover of Guyana, or the lover of the Amazon. And mostly these have been coralloid, especially in the past. This is a traditional concept for the genus that all of the species in this genus make coral-shaped mushrooms. But then we started to find these species of Clavulina that are highly reduced or restupinate. Sometimes we call these effuse. And we described 3 species from Guyana with this highly reduced morphology. So how do we know, one question is, how do we know these are Clavulinas? And there are 2 ways. So the first way is by microscopy. You can see that these basidia and spores are definitely Clavulina-like from what we've discussed so far. And the second is DNA. So we extract genes and build phylogenies, and we know from DNA evidence that these are in the genus Clavulina. And we can ask interesting questions with these specimens about evolution. How is evolution working in this group? In other words, how does evolution of diversity, how does diversity evolve? So one way we might think about this is that one hypothesis is maybe evolution runs one way, and everything that's complex comes from a very simple ancestor and it's always moving towards more complexity. But then we might encounter something that's less complex, possibly a step backwards and less intelligent, and we wonder, by the way, I want to say for my country, we're so sorry. But anyway, so sometimes evolution will run bidirectionally where we have an increase in diversity and then a decrease. And it opens a very interesting question, like how did this happen? How many other individuals like this are out there? What can we do about this? So the question at hand is, how does complexity evolve in fungi? We're going to use genomics to answer this question. So maybe it's that these simple, reduced morphology fungi are basal and they're older and they move towards more complex, or maybe it's that they're younger and they're more recently evolved. So to do this, we sequence genes and build phylogenies, which we can think of as a fungal tree of life. And it's not unlike a regular tree in that the trunk is the oldest, and as we move from the base to the tips, we have younger and younger individuals, and the closer the leaves are together, the more closely they're related. So we could think of a phylogenetic tree in the same way, where the basal branches are the oldest, and when taxa are closer together, they're more closely related. And so usually we read trees this way, and here I've mapped on the fungi with the more simple morphologies, and what you see is 2 places where these fungi come out, suggesting that this morphology has evolved at least twice in this group. So it seems like a gain of complexity for Clavulina, and then actually a loss at least twice in these lineages that we know so far. And I'll add that we have collected restupinate Clavulinas in other locations, and also Dr. Kania has collected some fungi recently around here that we're going to add to this equation. So another way of saying it in just plain English and backing out into the full order, which is the Cantharellales, is we start with very simple ancestors that have crusts or supinate morphologies. We move, oh, these got messed up, but that's okay. We move to more complexity with the genus Hydnum and the chanterelles, which are close relatives of Clavulina. Then we see an expansion of diversity in morphologies, especially in the tropics, and several reductions. So we have these resupinate individuals and a north temperate clade that remains coralloid. And we can add geography onto that. So here, these purple ones are all tropical and these ones are temperate, and begin to build a timeline. So far what we think is this genus, Clavulina, started in the tropics, diversified there, and then moved to the north and south temperate regions with these species complexes. And this is an ongoing project, so we'll have to keep working on this. I wanted to stop and acknowledge the people, the team that helped with this part of the project. So the PIs that got the funding for the project are Terry Hagel from Humboldt State and Kathy Aim, she's there, from Purdue. And we've had many great teams over the years in both Guyana and in Cameroon. And this is Noah Siegel, who took many of the photos that I showed you today. So if you would like to learn more about the project or see more mushroom photos, there are plenty available at tropicalfungi.org and also on my website, and my contact info is there, so please reach out if you feel inclined. And I'm going to save questions till the end, if that's okay. It's okay? So we just talked about, I'm going to switch gears now and talk about bacterial-fungal interactions. So we just talked about the direct interactions between fungi and plants. And of course we know that bacteria impact plant health in a direct way, both positively and negatively. But what we know less about is how fungi and bacteria interact and impact each other's health, functioning, and physiology. So this is a poplar tree. It's in the genus Populus. And we grow it in North America for alternative energy sources. So the Department of Energy is very interested in the poplar trees and the microbes that are associated for making paper and cardboard, and also for producing ethanol. So if we think of a poplar root, again underground, what we see is something similar, right? So here's our same root tip that's been colonized by a symbiotic fungus. But the complexity doesn't stop there. So if we zoom in on a single hypha or a single fungal cell, this is one fungal cell, we can see that there are actually bacteria living inside of these fungal cells. And we've sequenced the genomes of these organisms, and we see, if we zoom in on the bacteria and think about the bacterial genome, that the bacteria have been hit by phages, which are bacterial viruses. So there's this incredible amount of diversity and symbiotic diversity that enables us to ask how fungal bacterial interactions have evolved over time, and we're going to use this same systems biology approach. So again, the shapes will be in the corner to notate which kind of data we're talking about. So the big picture question is, how did these symbioses evolve over time? This is an obligate symbiosis, and we could think of this along a time scale. So these bacteria that live inside of these fungi are obligate symbionts. In other words, they cannot live alone. Their genomes are so degraded that we can't culture them. They've been inside of their host for so long, and I'll show you the data in just a moment, that they're on the far end of this spectrum. But anything that's in an obligate symbiosis started as two free-living organisms. So we can ask questions about how did these two free-living organisms come together and eventually over time form this obligate symbiosis. And there's a few things that I think have to happen along the way. So this is a conceptual diagram that just shows you they have to be at the same place at the same time. They have to be speaking the same language, talking in both directions. They have to figure out which resources or services they'll exchange for each other and when and why. And then with these intracellular endosymbionts, they have to get inside of the host. And this can lead to selection on the gene content that's important for the symbiosis. So, for example, if they're sharing food resources, like in this case they're sharing fatty acids, we can see selection on the parts of the genome that are involved in producing that product. Oftentimes in bacterial fungal interactions, those products are metabolites, and this can lead to metabolic cooperation. So by that I mean, it's a trade. Like you think you go to the market and maybe you make product A and I make B and we just trade wholesale, or maybe we collaborate and work together. So I'll do step 1 of the process, you do step 2, I'll do step 3, and what that leads to is gene activation and gene loss. So of course in these genomes, anything that's not being used can potentially be lost really rapidly and purged from the genome, and we see in these endosymbionts the signature of symbiosis and gene inactivation and gene loss. So again, this symbiosis is on the far end of the spectrum, but I thought I would just lay this out to give you an idea of how I'm thinking about these interactions. So here's a moment that I'm speaking really rapidly, and I said that I wouldn't, but here's a moment that I can stop and take a deep breath. And you all too are breathing in the audience, and part of the reason is because in your cells you have mitochondria. So these mitochondria were actually once free-living bacteria, and then endosymbionts, and now organelles, right? So this is a classic example of endosymbiosis becoming functional for a group, as well as chloroplasts. So of course, chloroplasts in plant cells were once free-living bacteria that got inside and are now helping their hosts. And this is a theme that we see with the fungal endosymbionts, that these bacteria have their own functions as free livers, then they move into these cells and offer their hosts a service that allows them to do new things. The other pattern that we've learned from the insect endosymbionts, or bacteria that live inside of insects, is that when these bacteria go into cells, they lose genes in patterned ways. So in this diagram, what you have are individual genomes as rows. These are individuals. And the genes are columns, and they're coded by function. So anywhere there's a black patch, these genes are missing. So if you zoom out, you look at the big picture, there's gene loss. It's not always the same genes, but it's always the same processes. And those processes are reflective of what they're sharing. So in other words, if the bacteria move into the cell and they're getting a product from their host, they no longer need to keep the gene for that product, and they throw it out. So you can actually read about the symbiosis just by sequencing the genome. So this gene loss is non-random. And then the third tool that we use is phylogenetics. So the idea is that because these interactions are so ancient, they're obligate symbioses that are co-evolved, we can actually make phylogenies of the hosts and the bacteria, and if we point them at each other, they'll have mirrored topologies. So for example, this tree is insects and this is bacteria, and because they have the same shape and branching pattern, this is an indication that these two groups have co-evolved together and likely as ancient. So we've been using these tools to ask this question: how do these fungal endosymbiont genomes evolve? Okay, this is a fungal phylogeny. It's to think of it as a fungal tree of life. And just before now, we were talking about the mushrooms, which belong to this group called the Mycota. And you may notice that these are actually quite young because they're on this short top branch. And there's the rest of a kingdom to think about. So the fungi that I'm going to talk about today belong to the Mycota. We used to call these zygomycetes. I know that you've seen these fungi before, so if you ever leave the berry or the bread out on the counter too long and they start to grow fuzzies, some of those are zygomycetes, okay? So we're talking about this group, the Mycota, and the bacteria, I'm sorry the font is small here, the bacteria that we're talking about are in the genus Mycoavidus, and they're sister to Burkholderia, which is pink in this phylogeny. So these are beta proteobacteria, and Mycoavidus is the fungal endosymbiont. Mortierella is the host, so this is the Mycota, and Mortierella elongata is the fungal host. Now, I just wanted to point out, in this tree, fungi that host endobacteria are in red. And in this tree, these are fungi that host bacteria also in red. So we don't have enough genomes yet, we'll sequence more, but if we take these trees and point them at each other, we start to see this pattern of coevolution where hosts of fungi are housing hosts of endosymbionts. Okay, so the punchline for this story is that these two organisms are really sharing fatty acids or lipids. So in this graph, you can see the bacteria living inside the fungal cells. And in fact, when we look, we always see them clustered around these lipid bodies. So Mortierella, this is Mortierella in culture, the fungus. It makes a ton of fatty acids, and they're pictured here. They have various double bonds in their carbon backbones that give them the kinks and the curls. And Mortierella makes all of these lipids, which it uses for its own physiology and functioning, but it also can trade those with its plant hosts and with the intracellular bacteria. And in fact, the bacteria rely strictly upon these lipids. So we're able to sequence the genome of both the fungi and the bacteria, and it's kind of a fun project because, like I said, we can't culture these bacteria, so we're growing the fungi in lab, and we extract DNA from the whole thing, start to assemble, and then we can use bioinformatic approaches to sort them into this is bacterial, this is fungal. So we have the fungal genome, which is here in several contigs, and here's the bacterial genome on one circular chromosome. So I've illustrated some of these are open reading frames, this is GC content, here's some functional genes, and you might be thinking this is like many other bacterial genomes, and it is, except for that it's highly reduced, which is this signature of bacterial endosymbiosis. So the question here is, how does this genome sequence reflect the dynamics of the symbiosis? And we answer this question by comparing the genomes of close relatives. So here's the conceptual diagram. Time is running this way. You have the free living Burkholderias in pink here, and then you have the obligate endosymbionts in red. And what we can do is compare the sizes and genome contents of these organisms to say, what happened along this transition? Again, you have two free living organisms and you move towards obligate endosymbiosis. How have the genomes changed over time, and how does that change reflect their interactions? So if we take, these are pie charts of just GO categories, gene ontology categories of what kinds of genes are in the genome, and we compare them, what we see is the following. So this is a free living Burkholderia. This one is a facultative endosymbiont, meaning it can move inside of fungal cells and then outside again, so it kind of does both. It has an intermediate lifestyle. And then this is an obligate fungal endosymbiont. Here, this is Mycoavidus and a close relative. So you can see, as time moves along, and we move from free living to facultative to obligate symbiosis, we have not only a reduction in the genome size, so here's the genome sizes, the genomes are getting smaller because of gene loss, but they're also getting different in their gene composition. So look, for example, this is protein degradation, this salmon slice. It's a small portion of this genome, but it's almost a quarter of this genome. So again, the idea is that these bacteria move into these cells, anything they don't need, they just lose, and we can use what they have left to infer what it is that they're getting from their hosts. Okay, this is a diagram of bacterial genome content, and it's kind of complicated, so I'll walk you through it. So here's the cell. Along the outside, I have illustrated all of the transporters. So you could think of these as what is possible for the cell to move in and out of itself. And then along the inside, you have various aspects of primary and secondary metabolism. So if we zoom in on primary metabolism, we have a couple of patterns emerging. So in this diagram, I've illustrated in blue gene products that are either impacted by gene loss or pathways that are impacted by gene loss. So I just wanted to point out a couple of things. These boxes right here are amino acids. You have a third of the amino acids in this genome are just gone. You need amino acids to build proteins to live, and these come directly from the host. So we know this because, for example, here's histidine missing from the bacterial genome, but it has this importer, so this is a product that comes, again, directly from the host. So we can use this logic, what's missing from the bacterial genome, what is it capable of importing to build evidence or data for the functional basis of the interaction between the two. And then the second thing, glycolysis, which is the derivation of acetyl-CoA from sugars, that's broken in 3 places. So most organisms rely heavily on this pathway for energy to drive the TCA cycle and other cellular processes, but this bacterium can't do glycolysis. So where does acetyl-CoA come from? It comes from the breakdown of those fatty acids. So the host is making these fatty acids. It has these importers for these products, and even in this reduced genome, there are several copies of enzymes in this pathway. So this bacterium is relying heavily on amino acids and fatty acids that come directly from the host. So it might not surprise you, given these strange gene loss patterns, that we can't culture these bacteria, and it's therefore hard to study, right? We have a genome sequence, and that's it, so we have hypotheses. And what we did instead was clear these bacteria from these fungi using a suite of antibiotics. So if you passage these fungi on solid to liquid media with a suite of antibiotics with different mechanisms, what you get then are, and we confirm this with our 16S primers, what you get are 2 isolates in the same background, the same fungal genetic background. One fungus has the bacteria, one does not, and you can compare them in different ways and say, how do these fungi behave differently when the bacteria are present versus when they're absent, and what does that reflect about their relationship? So here's what they look like. This is the same fungus. Look, the bacteria are in these cells, and here's the cleared culture. They look so different morphologically. So one is growing more aerial hyphae, it's looking different, but it's also growing faster. So So we can quantify the growth rate by simply tracing the colony on a plate. So you take a small piece of culture, put it onto a new plate, and every day you trace the perimeter and say, how quickly is this fungus expanding? So these cleared isolates are actually growing faster than their wild-type relatives, and we think that this is reflective of products that the bacterium is using. So in other words, there's a fitness cost for hosting these bugs. They're not taking a free ride. And the second thing that we notice is that they smell qualitatively different. Oh, it's too bad this video doesn't work. So they smell qualitatively different. One smells like wet dog, if anybody has ever known the smell, and the other smells a little less like wet dog. So when you have a dog and he's coming out of the river, the pool, and you get that smell, what you're smelling is the oils from his skin. They're being broken down by bacteria, the skin microbiome. So you're smelling beta oxidation, these products that are resulting from the breakdown of fatty acids, and we can smell that in Mortierella too. And we can quantify those products using a mass spec. So here I'm showing you, this is volatomic data. We have mass to charge ratios along the X, and these are quantifications along the Y. So in this graph, every peak is a product, and what I've done is offset the wild type profile in blue with the cleared profile in white. So anytime these peaks differ, like here's a white and a blue that are very different heights, that product is differentially abundant in the cleared strain. And so if we look particularly at products that are consistent with the breakdown of fatty acids or beta oxidation, we see more beta oxidation happening in the fungi with the bacteria. So in other words, we can smell the breakdown products that these bacteria are doing simply with our nose and with this sniffer as well. We've also looked at the intracellular level at metabolites. So now we're asking the question, at the cellular level, can we detect the breakdown products of these fatty acids? And here what I'm showing you is a fold change. So this is a ratio of each product in the cleared over the uncleared. So anytime they're negative, these products are becoming less abundant in the clear isolate, and these products are positive, they're accumulating in the cleared line. So all metabolites are along the X. They're organized by category here, and then I've highlighted in blue those that pass statistical significance. So the takeaway is that the fatty acids are the class that's the most impacted by clearing, and that in the cleared isolates, these accumulate up to 8-fold. So again, here's these bacteria that have the genes to break down these fatty acids. We can smell that process with our nose when the bacteria are present, and when they're absent, the products that they're using, these fatty acids, accumulate in the cells. Oops. So, but this does not give us an answer to the question, is this process direct or indirect? So in other words, the fungus could be doing something in response to the presence of the bacteria, or the bacteria could be using these products directly. And to ask this question, we moved on to a transcriptome approach, which I've illustrated in a computational pipeline here, but the important part is at the bottom. So one problem, which is actually not a problem, it's a joy of working with non-model fungal genomics, is that a lot of these fungal genomes have over half of their gene content is just unannotated, right? So you could do anything you want to get to your list of 100 genes, but if you don't know what those genes do, then how do you make sense of that information? So one approach that we've taken is by borrowing the annotation of the genetic model organism Saccharomyces and mapping on annotations from that genome to our genome. So from now, I'll talk about genes that are differentially regulated, transcripts that are differentially abundant, but only those that have clear orthologs in the Saccharomyces genome. So we can see the signature of the sharing of these fatty acids. We see that those genes are differentially regulated in the presence and absence of these bacteria. But I wanted to share with you a piece of unpublished data, which is these genes that fall under the gene ontology class response to chemicals. So here what I'm showing is a pie chart of just the classes of genes that are differentially regulated in the clear isolate. So this doesn't have information about whether they go up or down. It just simply means which genes are affected by this process. So the first two are carbohydrate and lipid metabolism. And I'm going to skip those for time's sake, but this third one is response to chemicals. And what actually ends up falling under this category are genes that are involved in mating in Saccharomyces. So in Saccharomyces, which is an ascomycete, they sense their mating partners by using these GPCRs. These are membrane-bound proteins. You could think of it as a switch or a sniff. So here comes a pheromone, or like a mating pheromone, a signal that a potential partner for mating is nearby. And once that signal is transduced to the inside of the cell, there's a signaling cascade that tells the fungi, okay, now it's time to turn on another transcriptional program for mating. And what we see in the cleared isolate is that several genes in this pathway, which I've colored green regulation is up and red is down regulation, several genes in this pathway are differentially regulated in the cleared isolate. So we have an internal subunit of this structure. This is the brakes on the system. It prevents signaling in inappropriate situations. So in other words, the brakes are off. And this is a pheromone pump. So what I think is happening is these endosymbionts may have a role in the potential, like the assessment of the proximity of a suitable mating partner. So I think there's a link between endosymbionts and mating. So one question is, what aspects of fungal GPCR signaling are mediated by these endosymbionts? And this is a little more broad because GPCRs are involved in many processes. So mating is one, lights, sugars, amino acids are several others. So I think it's clear that some aspect of GPCR signaling is affected, but it's also, I think it's possible that it's mating for the following reason. So the zygomycetes, like I said in the beginning, are early diverging fungi. We're just learning about them and sequencing their genomes. And most of them sense their mating partners in the following way. So you have spores that germinate into hyphae, and these individuals sense each other by the production of trisporic acid. So these are intermediates of the trisporic acid biosynthesis pathway. So if one isolate makes this intermediate and one makes this intermediate, then they can mate, and this is the cue for them to form these sexual spores or zygospores. But what's curious is that in the Mortierella genome, these genes are not present. So it's clear that they do sex because they make these sexual spores, but we don't know how because they don't have the traditional machinery. So another way of saying that is these younger, earlier diverging lineages sense their mating partners through GPCRs, and then the more basal Mucoromycota use these trisporoids, but Mortierella is anomalous because it doesn't have the genes for this pathway. So how it's having sex, we're not quite sure. So one question is, how does Mortierella sense its mating partners? And it seems like, based on the transcriptome of the cleared and uncleared isolates, that endosymbionts may have a role in this process, which would be, it would have huge evolutionary implications, right? Like if you have the potential to outbreed and bring in new diversity into your genome, and some do and some don't, those are testable hypotheses. As well, this hypothesis is well supported in the insect endosymbionts. So I told you that many insects have bacterial endosymbionts. Yeah, and I see some shaking heads. So, for example, Wolbachia is a bacterial endosymbiont of insects that has control over the host reproductive cycle and can actually dictate which other insects it's capable of mating with. So in other words, it has a role in the future diversity of the population. So with that, I thank my collaborators. I have a ton of great collaborators from Duke, including Dr. Camille. And of course at the Joint Genome Institute and the Oak Ridge National Lab. And then these are my collaborators to the University of California system on a project I didn't have time to talk about today, but they definitely have helped me along the way. So I thank you for your attention, and I welcome your questions if you have any.
Thank you very much, Jessie. ¿Tenemos tiempo para preguntas?
Hello, thank you so much for your talk. It was really interesting to see how cool the fungi that Camille is studying are. I was wondering, what's the coverage you have for these genomes that you're studying?
Yeah, it's a good question. So do you mean like are there artifacts in the assembly for the sorting, or?
Well, what I'm concerned is if there are artifacts and you're not finding all the genes that you say are actually missing.
Oh, for the bacterial side. Yeah. So that's a good question. So we've done this several times, and the first pass, what we did is take the assembled contigs and sort them into bins based on homology, just a blast score, like this is bacterial, this is fungal. And then we had fragmented genomes. So you're right, it's hard to say a gene is lost if a genome is incomplete. However, then we got some longer reads, some PacBio reads involved, and we're able to close the circular chromosome, so we're confident that the genes that are lost are actually missing. And we're starting to branch out now from one isolate into relatives, so we have a couple endosymbiont genomes, and we can say, indeed, this gene is lost in multiple individuals and lineages, which I think helps a little bit. But you're right, you can't be sure about gene loss without a complete genome sequence.
Have you thought of maybe doing a simple PCR and trying to find that gene in the
Design primers across the span and say, is it truly there?
Yeah, I mean, they should be there, right? The priming sites, and if you have a negative result, but then a positive one that does have it, then you will confirm.
That's another good suggestion. So we've looked a little bit in work that I did not present today at proteomics of the endosymbiont fungal interaction, and we can see in certain conditions bacterial proteins turning on, so we feel confident that the gene is there. Yeah, that's a good question.
¿Más preguntas?
Okay.
Are there any other Mortierella species without the bacterial?
So it's a complicated answer. So many Mortierellas have bacterial endosymbionts, and more frequently, so this one is dominated by Mycoavidus, but more frequently there are multiple co-occurring bacteria from different lineages. So many proteobacteria, some firmicutes. You could almost think of it like a fungal microbiome. And that varies based on the isolate. So some have beta proteobacteria, some seem to have less endosymbionts, some have more diversity. But I will tell you that almost all Mortierellas, many zygos, and in fact many ascos and basidios have been detected recently with intracellular endosymbionts. So I think this is an aspect of fungal diversity that's underappreciated. I think the more we look, the more we'll learn about these endosymbionts that we couldn't detect until we had sequencing capacity for genomes. Yeah. We have another one?
Hi, thanks for the talk. It's great and it's a fascinating topic. So I have a couple questions. One is, where did these bacteria come from? Like, how are they inherited, or is it all vertical, or is there evidence for some horizontal transfer?
Oh, it's three separate great questions. So so I think the bacteria, well, I'll say this, their closest relative is Burkholderia, and there are many free-living Burkholderias in the rhizosphere, for at least a poplar for sure. So I'm thinking that they got into the rhizosphere with plant interactions and then came into the vicinity of the fungi, somehow entered the fungal cell. We're not quite sure how. There's a sister, like a relative system, where they figured out some bacteria will exude chitinolytic enzymes, so to physically punch a hole in the fungal cell wall to get in, but I don't know how these ones got in. And then as far as transmission, I know that it's at least partially horizontal because I'm able to, in lab, take a culture, take a small piece of the growing culture and put it onto a new plate and still detect the endosymbionts, so they're passed through the filamentous hyphae. But I've also done some unpublished work looking at bacteria in the spores, so you can make asexual spores or zygospores, and we can detect that the bacteria are also packaged in the spores. So I think it's both vertical and horizontal. And I would guess that, we've done some dating in the phylogenies, and this relationship is very ancient, and I would guess, I don't know, but I would guess that it would be hard to maintain that symbiosis over the years if it didn't go through spores or, It's a good question, though. It's got predictions for the way that evolution could work.
Well, the whole coevolution premise, sort of, based on
Yes,
the premise that it's vertical, right?
Yes.
Yeah. So, and the other question is about whether you've looked at, you have this great system where you can basically play with both layers, right? The host and the symbiont, and then look at whether, you showed some evidence suggesting that the host benefit, well, there's costs associated with hosting a bacteria, but also that the bacteria might be involved at some sort of sexual compatibility system, right? But have you thought about looking at other potential benefits? Say, I think this is like a great system to look at ecological breadth and look at whether, the associate brings in some other benefits that are not necessarily just metabolic low mix, but actually looking at the more of the ecological side of it.
Sure, yeah. A couple of things. So I think it would be hard to maintain, so we showed a fitness cost, but the settings that we're looking in are in monoculture on rich media in lab, which is in no way reflective of the soil environment, right? Like it has no competition, nutrients are not limiting. So I'm thinking that some of the benefits may actually be contextual. And that so the interesting thing about Mortierella is that it has no, this is a hypothesis, I don't have any data for this, okay? So the interesting thing about Mortierella is that it has no genes for the production, almost none for the production of secondary metabolites in its genome. But the bacteria has plenty, and they both have transporters. So I'm thinking housing these bacteria, even though there's a cost, like a trophic cost, it may make you competitive when you have other species around you. So in other words, we can't detect it in lab with this method, but you're right that there should be a benefit somewhere if this is how we're thinking about it. Or maybe not, maybe it's just a parasite. Yeah, that's a good question.
Mortierella is one of the most abundant fungi in soil
Yes,
all over the world.
yes.
So it's very intriguing to me what you say that they don't have metabolites because they have to compete a lot and they have to be very good competitors in the soil.
Yes.
So maybe experiments of making different strains of Mortierella with different bacterial lineages competing between them and also with other soil fungi. Maybe these bacteria are very important in giving these fungi this ubiquitous capacity of explore all the soils in the whole planet.
Yeah, I like that idea. It's a good suggestion for an experimental follow-up. I'm also thinking of Mortierella grows so fast. I think that's one of its strengths is that it can outgrow its competitors. But I do think that there's some legs to this idea about secondary metabolism. Yeah. It also has a bunch of chitinolytic enzymes, which I think gives it a competitive edge in some situations. It has access to a resource pool that many organisms cannot degrade.
Also, they use very simple sugars, so
Yes,
they compete with.
We heard them on malt, so yeah.
¿Alguna otra pregunta?
Okay, I have more some kind of maybe philosophical questions.
Okay.
When you look at these interactions, so how did you really determine that this is naturalism and not parasitism? Like from the
Yeah.
bacteria?
So the data that we've generated suggests strictly parasitism, right? We have this fitness cost, we've attributed it mostly to fatty acids and possibly amino acids, and what is the benefit? And I think that my hypothesis is secondary metabolites, it's testable. We should collaborate on it. Yeah. If there's a benefit in another setting, I think it could be potentially, like if you can control the sexuality of your host, then I don't think it's necessarily a benefit for the host, which would make it a parasite, right? But you could keep your house intact from the bacterial perspective, and that might be part of it. But I think with all the changes in the volatile, so we can smell the fatty acid breakdown products, but there are a ton of other products, and we know that volatiles are very important for plant-fungal interactions. So I think there are ecological repercussions for these fungi when they have endosymbionts and when they don't. Like they really make their host function in a completely new way at the transcriptome, at the proteome, at the metabolome, like all these different levels. So I think that there may also be bigger picture, we're looking in a really reduced system. There may be changes or benefits in a different context that we haven't considered yet, but yeah. Also, I think philosophically there's a scale between parasitism and mutualism that's kind of blurry. Like you said, you're a parasite and you're a mutualist and they're separate, and I think in reality you might be able to like slide back and forth, but it's testable. Yeah, great question.
Tenemos tiempo para una última pregunta.
I have another philosophical question,
Yeah.
and it's about if these organisms are obligated inside all the time they hyphae, why they are not considered as organelles?
That's a great question. So I think that they are probably on their way to being organelles. Within the insect endosymbiont literature, there's a debate whether, okay, so you're a bacterium and you get inside the cell and you begin to undergo gene loss, does that process stop and you reach an evolutionary stasis? Or does it continue and you just degrade till you're gone? What is the balance of power between the host and the endosymbiont? And I think that's fascinating. I don't know, actually. One place that we haven't looked that I'd like to look are at genes that control DNA repair. So sometimes if you lose like DNA repair genes, then you're in sort of mutational chaos and you might degrade into oblivion faster. But that's another fantastic idea. I think we need more genomes. We can test all of these.
Bueno, les agradecemos mucho su asistencia. Los invitamos a tomar café, galletas y papitas de este lado. Si tienen más preguntas para Jessie, es el momento. Thank you very much.
Thank you guys.